and I've learned a lot. I wanted to talk about our efforts towards making a pan-sarbecovirus vaccine. That is something that would we're hoping would protect against the emergence of a new sarbecovirus, that is SARS-like beta coronavirus. So I wanted to start by reminding everyone that this is the third epidemic or pandemic we've experienced in the last 20 years. So we started in the early 2000s with SARS-CoV-1, it's now called, and then we had MERS in like 2012. Both were epidemics, which are either gone or under control, but now we're in the middle of SARS-CoV-2. And of course, there are 4 other common cold coronaviruses that cause common colds that circulate among humans, but don't cause a lot of significant health problems. But the point is that these arise in animal hosts of some sort. And I think we're only seeing the tip of the iceberg. So when I started working on coronaviruses at the beginning of the present pandemic, I read the literature, and everyone who worked on coronaviruses said it's only a matter of time before we have another epidemic or pandemic. And sure enough, SARS-CoV-2 came. So I think we need to be prepared ahead of time for another spillover event. We've been lucky so far. Sarbecoviruses are known to spill over into humans who live, for example, near bat caves in the province of Yunnan in China. They just don't go on to infect other humans usually, but as we know, these viruses can have many variants, and we only know the tip of the iceberg in terms of their sequences. Okay, so that being said, now I want to remind everyone about the viral life cycle. It's really, coronaviruses are just like any other virus. They enter the host cell through some kind of receptor. They make copies of themselves, and they release new viruses from the cell to infect other cells in the same host or infect a new host. In the case of coronaviruses, they have the famous spike on their surface, which then binds to a receptor on, for example, that lines the epithelial cells of the lungs and other organs. So that's ACE2. If you have neutralizing antibodies that are raised against the spike, you can prevent viruses. This is a typical IgG here. And so if it binds to the spike and somehow prevents a productive interaction with ACE2 by some sort of mechanism, you'd prevent the subsequent events. So that's what people are trying to raise, for example, by vaccination. There are, of course, other mechanisms of protection as well. Okay, but I want to concentrate on neutralizing antibodies here and remind people that the spike trimer structures of coronaviruses look like what you're seeing in the lower left there. And they have these domains called receptor binding domains that can be in either an up or a down position. So they're able to actually fluctuate between different positions. And this has been revealed in structures of SARS-CoV-2 coronavirus spikes, and prior to that, other coronavirus spikes. So I just wanted to show you in this movie, this is a surface representation of a SARS-CoV-2 spike. The other coronaviruses spikes look the same. This is now a cartoon diagram showing the alpha helices and beta strands. That's what it looks like with the 3 RBDs in the trimer down. Here's one RBD in an up position. And now you're going to see what it looks like if all 3 RBDs are in an up position. And now you're looking at the spike trimer you're going to see it from the top to look at that transition between the closed conformation, all 3 down, and an open conformation where the RBD can be up. So that's what's meant when people talk about up RBDs. The RBD has to be in an up conformation to bind ACE2, but that up conformation is not triggered by the binding of ACE2. It exists in an equilibrium between closed and open states. And then antibodies can recognize either closed or open RBDs. And we've been concentrating mostly, but not exclusively, on antibodies against the RBD. That's a major target of neutralizing antibodies. Okay, so early on in the pandemic, we meaning Alex Cohen, who was then a graduate student in my lab, used what we call a Mosaic nanoparticle approach to try to make a potential pan-sarbecovirus vaccine. So again, this means SARS-like beta coronaviruses. So in summary, this is summarizing the work that he published about a year ago. So basically, he selected receptor binding domains from SARS-like beta coronaviruses that were known to be of pandemic concern. I'll show you which ones he used in a moment. He then attached them all at once to make what we call a Mosaic RBD particle. And so this is a nanoparticle we're using, which I can address questions if anyone has it about how we do this, but we're able to attach variable numbers of RBDs to a single nanoparticle, which has 60 attachment sites. So there are, in this case, 8 different RBDs here. He then did studies in mice, and I'll show you follow-up studies we've done in other animals as well. And what he found was cross-reactive antibody responses that looked very promising. So why do we want to make this Mosaic vaccine approach? Like, what is the purpose of this? So this is our hypothesis for why this might be a good idea for making, focusing the immune response, the antibody response on conserved regions of an antigen in general, and in this case, conserved regions of an RBD. So here's schematically what you imagine might be happening on B cells that have membrane-bound antibodies. So that's their B cell receptor. So there are a variety of these in germinal centers, for example, B cells presenting different types of antibodies as membrane-bound B cell receptors. And if you injected homotypic nanoparticle, which is one in which every antigen is the same, what you're going to get is a lot of antibodies against what we'll call strain-specific distracting epitopes. They're represented by the orange triangle there. And so because you have cross-linking between adjacent antigens here, the distracting epitopes are right out there ready to be bound. And so you get binding of B cell receptors, even if they have intrinsically low affinity for the antigen, because they can cross-link between adjacent RBDs. So now let's say instead you injected a Mosaic nanoparticle where the distracting epitopes are still there, but there's a low probability that you'd have the same distracting epitope represented on adjacent antigens. So here you see distracting triangular epitopes, but you don't have 2 orange ones next to each other, or not very often. And so then you would presumably not trigger as many of these orange types of antibodies, which we don't want. But instead, let's say that what you want are antibodies against the purple epitope, which is more conserved. And what this would mean would be, let's say the purple epitope is somewhat sterically hindered, so it's kind of hard for antibodies to bind to that region. But if you have cross-linking there, you can increase apparent affinity through avidity effects. And so you might trigger preferentially these purple types of antibodies. So what do we mean in terms of structurally about the orange regions and the purple regions? So what I'm going to start with is follows very nicely from Christian Gabler's really great talk earlier today. These are the structural studies we've done in collaboration with the Michelle Nussenswag's lab at Rockefeller University. And as you heard, they began to collect samples from COVID-19 convalescent donors and later from vaccinated donors and so on. And so early in the pandemic, we began collaborating with them to solve cryo-EM structures of spike trimers from SARS-CoV-2 bound to these antibodies. So what I'm showing you is a summary of a lot of different structures. So this effort was spearheaded by Christopher Barnes, who was then a postdoc in my lab. He's now an assistant professor at Stanford University. He started there in last July. And I'm not showing you all the structures he and others have solved in my lab or around the world. We've solved probably at least 60 of these. And we're seeing general themes here that I want to emphasize by just showing you a few of these structures. So the first thing I want to point out is that in this slide, the name of the antibody is up at the top, and Christian already introduced the classifications, but I'll go into that a little bit more. But what we're doing here is we're looking on the spike trimer structure at whether or not the antibody binds to an up versus a down RBD. And some antibodies can bind to either up or down RBDs. Some antibodies like C144 can only bind to down RBDs. And so we know the structural reasons for all of this from the in-depth analyses of these structures. So these are analyses in one of Christopher's papers that looked at antibodies from the 1.3 month COVID-19 donors that Christian described, and these were described in Davide Robiani's paper, which is right there. Okay, and then later, as was described, Vinci Wang from Michelle's lab isolated monoclonal antibody sequences doing single B cell cloning from vaccinated donors, and we solved a bunch of other structures, and basically it's the same type of antibodies in terms of their structural recognition of the RBD in the vaccinated versus the infected donors. So there's nothing really different going on here. So there's a lot of in-depth analyses in the structures that I really won't go into, but I want to just show this slide that Christian also showed, which was our attempt in November of 2020. This paper came out where we classified RBD binding antibodies into these 4 classes. And these are based on both their epitopes, but also based on how they bind in terms of can they recognize up versus down RBDs. So we're taking the RBD structure from the trimer structure. So all you're seeing now is an RBD monomer, but really these are from structures with spike trimers with antibodies bound. And so we classified these into 4 groups. It's very clear there are more epitopes than this, and antibodies that I'll show you an example of that kind of cross between these sorts of classifications, but we find this a useful way to classify things. Other groups have different ways of classifying them, but this is ours. So what we did here was to list antibodies at the time from our studies and other studies that fell into these categories. And I want to point out that the class 1 and class 2 so-called anti-RBD antibodies are in this depiction are up at the top of the RBD, and this is actually against the more variable part of the RBD. I'll come back to this a lot. Whereas the class 3 and the class 4 antibodies are more directed against the part of the RBD that remains more constant. And so among sarbecoviruses, and also that's the part of the RBD that doesn't vary as much in the mutations that are found in the variants of concern, including Omicron. And I just want to point out that some of these, antibodies right here were used therapeutically. For example, one of the Regeneron antibodies, the Eli Lilly antibodies, and these worked very well, but now they don't work against Omicron, which has a lot of mutations in this part of the RBD, whereas an antibody from Vir, which binds more down here, remains effective against Omicron. Okay, so this is from our paper a year ago in November, where we have this large table which has a lot of information we tried to compile about the antibodies, the structures that we had solved, and the structures that were available at the time. And I think a lot of the conclusions are still true, even though there's more structures by now. But the point is that if you look at one thing that Christian brought up is that this is the number of amino acid somatic hypermutations. There's not a lot in these antibodies. They're practically germline. This has been noted by a lot of different studies. And it's very interesting that you get antibodies that neutralize at very low concentrations. In other words, single-digit nanogram per mil sometimes, even though they're barely somatically hypermutated. But I wanted to point out that this class 4 site, which people knew about early on because of this CR3022 antibody that was isolated from a SARS-1 infected person and cross-reacts with SARS-CoV-2, they're not generally as strongly neutralizing. But this is what we actually would like to target, what we think we're targeting in our vaccine. And so let me show you now the structure of the RBD with the variability between sarbecoviruses plotted on it to it. And so we've got a gradient. We take the sequences of RBDs from sarbecoviruses. We have a gradient from orange to purple, where orange is the more variable regions and purple are the less variable regions, and you're seeing the RBD surface structure from 2 different orientations. And you can see that this top part where you have class 1 and class 2 epitopes varies a lot more. And again, that's the place where the variants of concern have mutations for the most part. And then class 3 and class 4 are more in these purple areas. So we'd like to target class 4, but I just said they're not that strongly neutralizing. So wouldn't that be a problem? Well, it's not, because you can raise antibodies that span between, that actually they're class 4, but they reach up towards the class 1 site. So you might call them class 1/4, but they actually block ACE2 binding. And so they're more potent against in neutralization. And so these are examples from the original Robiani et al. Study. So COVID-19 infected donors do raise these antibodies. They're just not very prevalent. So what they tend to raise, and vaccinated people tend to raise, are the antibodies against these orange regions. So this is what we're trying to do. We're trying to target the base of the RBD to get this epitope that is more conserved, or to get these regions of the RBD that are more conserved. So here's the RBD in the 3 down positions. And now we're going to show you one of the RBDs in an up position. And now we're going to plot the orange and purple parts onto that. And you can see that this purple part is less accessible in the context of the spike trimer. But if you could access it and you could preferentially target it, you might get antibodies like we know can be raised by both vaccination and by natural infection that target this region, but also block ACE2 binding. So that's what we're trying to do. Okay, so are we doing that? So let me just quickly go through what was published in Alex's paper. So basically he made Mosaic, he made homotypic nanoparticles that only have SARS-CoV-2 RBD, so 60 copies. Then he made Mosaic nanoparticles that either had 4 different RBDs, or Mosaic 4A, Mosaic 4D had these 4 RBDs, and then he made a combination that we call Mosaic 8 that has both a 4A and 4B. And now what I want to point out is we deliberately left 4 RBDs off of these particles because these represent mismatches. So we can look in binding and neutralization assays to see if we're getting responses against SARS-CoV-1, for example, and other RBDs from sarbecoviruses. Okay, so this is a summary of data that's both published and unpublished. Here we have comparisons of ELISA binding titers of mouse sera after a prime and a boost. And you can see here that the better responses are more in these reddish colors, the worst ones are green. And so you get cross reaction if you inject the homotypic particles. They work really well against SARS-CoV-2, but you get cross reactions to these are the mismatched strains here for the Mosaic. But the homotypic also works. It's just that it doesn't induce as high binding responses against the mismatched strains as the Mosaic does. So that looks good for the Mosaic. Now, we also started an NHP study, that's non-human primate study, that is, where we've only looked at Mosaic 8 injected animals, and that's ongoing at Bioqual. And these animals were challenged with either SARS-CoV-2 or another cohort with SARS-CoV-1 in late November of this year. Okay, and then we also see neutralization by both the mouse sera, and that's published. And so you can see better responses when we do the Mosaic 8 versus the homotypic comparison against the mismatched strains. And we're seeing good responses in the NHPs as well. So I wanted to say that we have some evidence that we're actually targeting the epitopes that we want. And this evidence comes from the cloning of monoclonal antibody sequences from Mosaic 8 and homotypic injected mice. This was done by Miso Park and John Williams at the City of Hope. So what they were able to do using a Berkeley Lights beacon instrument, so they could do this very, very quickly, was isolate B cells that secreted IgGs that bound to 4 different RBDs. And so we don't have a lot of these, but we do have 5 monoclonal antibody IgGs that we've expressed and analyzed from Mosaic 8 injected mice. And then they also were able to get 2 monoclonal antibodies from the homotypic immunized mice that bound, the criterion was you have to bind to 4 different RBDs. And so what we did then was we expressed these and we did ELISA binding against SARS-CoV-2, SARS-CoV-1, and some other sarbecoviruses, and also the variants of concern at the time. And so you can see that we've got really good cross reactions for some of them, not all of them, but these 2 over here look really good in terms of cross-reactive binding and cross-reactive neutralization. So far, the monoclonals isolated from the homotypic injected mice aren't quite as good, but we need a lot more examples to really conclude anything. So what we're trying to do here is we're trying to use an iterative approach where we inject model organisms. We look at the polyclonal response and the monoclonal response. And what I'm going to tell you about are, and then we do neutralization assays for both, but we also want to look structurally at the monoclonal antibodies to see where they're binding. So what epitopes are they actually recognizing on the RBDs? So of course, this would involve a spike trimer structure where we then localize where the antibody is. And then what we're going to show you are actually where the epitope is on the RBD alone. And so Cheng Fan, a postdoc who joined my lab about 6 months ago, has already solved, well, she's probably solved 30 or 40 structures, but she's solved all 7 of the monoclonal antibodies bound to the spike trimer that I showed you before. So here are their epitopes. This is for reference. This is the ACE2 binding footprint. So the receptor binding footprint is in that orange part. And then these are the binding epitopes for those 7 monoclonal antibodies. And you can see they're targeting, for the most part, this purple region. Note that this antibody here does not target the purple region. And it was clear from the binding response that that was a strain-specific antibody that was mostly going for SARS-CoV-2. So it's not that we're not getting those, but I think we've skewed the immune response to hit the more purple regions. And I want to point out, just for the for Omicron, these are the orange places are where there are Omicron substitutions in the RBDs, and you can see that they're just not in this class 4 region. So if we're hitting the class 4 region, it's hopeful that we're not going to have problems with not working against Omicron. And that's borne out by our NHP study, which is ongoing. So we're doing polyclonal antibody. These are just ELISA binding titers. The neutralization assays are ongoing, but we're not seeing really any diminution against Omicron after the boost. So that's Omicron there over here. So compared to Delta and Beta and the original strain, it's the serum antibody binding response is the same.
Pamela, it's the 2-minute warning.
Okay, so we just got these data actually last night from Bioqual. We're seeing protection against the SARS-CoV-2 challenge. The SARS-CoV-1 challenge was done on the same day. It cannot be analyzed right now due to a variety of rules in the United States against SARS-CoV-1 studies because that's a select agent. So we're trying to work those out. But so far, so good. We're seeing what looks like reduced infectious virus in bronchial alveolar lavage and also in nasal swabs. Okay, but this is a study that could be done at the Rocky Mountain National Labs by Neeltje van Dormelen and Vincent Munster. And so what you're seeing is a human ACE2 transgenic mouse model where the mice die if they've been infected with either SARS-CoV-2 or SARS-CoV-1. So what you're seeing is that the Mosaic injected mice do better. They do as well as the homotypic animals that were injected in terms of a SARS-CoV-2 challenge. And this Mosaic doesn't work quite as well, but this is the original Mosaic here. So the red and the blue are the Mosaic and the homotypic, and this is the survival curve over here, and the control animals die. But the more exciting thing to us is that what works in protection against SARS-CoV-1 are the 2 Mosaic injected, those animals that were injected with the Mosaic nanoparticles. These survive and they do not lose weight, but the homotypic animals actually die. So we think this is proof of principle of what we're trying to do. So this is my summary. So basically, when we multimerize the RBDs on these nanoparticles, we get increased immunogenicity, as you'd expect from basic immunology. And the exciting thing for us is that when you co-display just a SARS-CoV-2 RBD in the presence of 7 other RBDs, it works just as well as the homotypic, where all of them are from SARS-CoV-2, to recognize and neutralize and protect from SARS-CoV-2. So we think this means that this would work now for a vaccine against SARS-CoV-2, but then it also would have advantages for protection against mismatched strains. So here where SARS-1 is representative of a mismatched strain that might be similar to something that could emerge from a zoonotic source, and we're seeing binding, neutralization, and protection in the K18 transgenic model against a mismatched strain with the Mosaic injected animals. And also for these particles will survive lyophilization. So if they were a vaccine, there wouldn't be a cold chain storage. So taken together, we think a Mosaic nanoparticle approach would be a great way to protect now against SARS-CoV-2, to protect against SARS-CoV-2 variants of concern that are here now and that will emerge in the future. And then importantly, against something that might emerge in the future, another sarbecovirus spillover event. So I just want to thank everyone. I didn't talk about HIV, but the coronavirus structural studies were started by Christopher. He's now at Stanford as a professor, and Cheng Fan and all these other people are working in various ways. And Alex Cohn led the Mosaic nanoparticle approach. So I'll stop there.
Thank you so much, Pamela. And again, I'll just remind folks, if you have questions, please go ahead and raise your hand and we'll allow you to speak. And I'd like to lead off with a couple of things if I could. The first is maybe it's odd, I'm not sure. Early in the pandemic, there were studies suggesting that some of the differences in clinical presentation from no symptoms to death may have been due to prior exposure to some of the more common human coronaviruses. And I'm just wondering, if you were to look at the RBDs from the common cold coronaviruses, they still have this purple region that you're talking about as being targeted, or is it something different?
No, the RBDs so first of all, in Christopher's first paper in 2020, we looked at the convalescent plasmas for cross reactions with common cold coronavirus spikes, and we didn't see them. We just didn't see that in those SARS-CoV-2 infected people. So I don't think that would be due to antibody responses. There are antibodies that could cross react, for example, down on the S2 region near the fusion peptide and so on. But I suspect if there's any protection from common cold coronavirus infections, it would be at the level of T cells, because you have peptide epitopes that are going to be the same. But in the RBD, our vaccine approach doesn't work against MERS, which is a lot more closely related to sarbecoviruses than common cold coronaviruses are. It's another beta coronavirus. So I don't think this will work as a pan coronavirus vaccine. I'm usually very careful to say that.
Got it. And with respect to the nanoparticles, 2 questions. How did Alex choose the 8 different RBDs to present? And then do you know whether there's a difference between immune response in animals for the nanoparticle versus, say, a mixture of RBDs just injected freely, something like this?
Okay. So how he chose them was this paper from Vincent Munster's lab at the Rocky Mountain National Labs had just come out, and they took all these sequences known at the time of sarbecoviruses that were thought to be of pandemic potential. And we chose them based on that paper and also some of Ralph Barrick's studies showing, that WIV1 and SHC014 were thought to be of pandemic potential. And then we also tried to spread them out across clades of sarbecoviruses. So that's how we chose them. And then we deliberately left out clade 3, RBDs from clade 3, because those are more distant. So we wanted to use those for the mismatches. And what was your second question? Oh, yeah, a mixture. Yes, I mean, maybe that would work, but we're accumulating evidence here that this approach here relies upon tethering the antigen to some surface. So mixtures of RBDs that are not tethered to anything are not going to be very immunogenic. I mean, they will work. Peter Hotez's RBD vaccine is just RBDs in alum. It will work against SARS-CoV-2. I doubt it will give some cross-reactive responses because you will make antibodies against this purple region. But we believe that you're going to target your response more to the purple region if you do the Mosaic approach. So that would include also making, collections of homotypic nanoparticles, each of 8, let's say you make 8 homotypics and inject those as a mixture versus making the Mosaic nanoparticles. So we're in the process of just nailing that down with a lot of animal studies. But I think so far we're really getting promising results that we are targeting that purple region, which we're trying to get.
Perfect. Thank you.